Okay, well, it looks like it's time, and everybody's back from the coffee break, which is a good sign. So we didn't lose too many people. So this is a talk where we want to talk about how the use of Boolean models can be utilized to model dynamic behavior. And we are going to talk today more about gene regulation. And then for the level 2 lecture, I have some ideas that will include much more on signal transduction. But they are going to be quite similar in nature, the mathematics for those two topics. And we'll do it as we always do it with Robin. So I'll talk where the mathematics needs to be explained. And when we get to the biology, Robin will say what needs to be said, because I can probably pass for having some biology knowledge in the train, but this is not the venue to try this. So again, my kind of wonderful blue ball machine here.
It's overly simple.
This yeah. So again, the level of detail. So why do we want to talk about Boolean models? Because if we want to use continuous models, the level of detail at which those systems generally need to be represented in the model is huge. And as much as we know about biology, we do not know all of the details. And again, that's going to be a recurrent theme. So we have questions that are huge for which we only have partial information. So how does the system behavior relate to a network structure? So if I show you how the variables in your system interact as a graph, then can I, from this graph, tell you what that system is going to do on the systems level? That's a huge question. Can I break this system, which nowadays we want to study systems of hundreds of thousands of 10,000s of variables and nodes? Can I break it into simpler systems, study them independently, and then kind of put them together, understanding what the regulatory elements are? The context in which those examples are important: signal transduction, gene regulation, huge questions there. And mathematics seems to be capable of answering some of those questions. So just a little bit of biology, to the extent I can handle it, that at the molecular level, the systems function using two types of information. You have the genes that encode the that encode the information and execute all the functions in a living organism. And you have the interactions in between and the signal transductions that facilitate those. And for qualitative models, we can actually try to do things with zeros and ones. We can say, all right, I have quantities that are considered either present or absent. And at first this bothered me quite a bit, because I say, well, how come, right? I mean, where do you draw the threshold? But in gene regulation, I've learned, you may have trace concentrations of certain enzymes at all times. But when the gene is on, those concentrations change by thousands of times. So they will increase 1,000-fold. And so in that sense, those trace concentrations could be considered zeros, and then the much, much higher concentrations could be considered one. So that's not such of a stretch. And of course, as soon as we introduce Boolean in mathematics, we immediately think of Boolean logic and the operations of and or and not. And these are the notations that I'm going to use for those operations. So more for the biologists here in the audience, that we say that A, B, or C are Boolean variables when we mean that we don't know what exact value this variable may take at each time, but it generally can take values 0 or 1 and nothing else. And then we can construct so-called compound Boolean expressions where we use the Boolean logic operators and or. So this is A and B. This is A or not B, the opposite of B. So if B is 1, the opposite will be 0, and vice versa. And then we can compound those and make more and more complex expressions. And as with the multiplication tables, we have the tables for those operations. They're much smaller here because we only have 0 and 1 as options. So notice that and is 1 only when both A and B are 1, and or is 0 only when A and B are both 0. So in that sense, and is important when we talk about signal transduction or gene regulation. And is important when you need input from both nodes. So this and that. So both are needed to regulate another node in the network. Whereas using or would mean one or the other or both. And then we can evaluate those expressions based on the tables for those operations. So here is, for instance, an example where if we want to evaluate this expression for those particular values of A, B, and C, we can do it, and we do this following, of course, the order of precedence. So parentheses trump everything. Then negation has the highest precedence after that, then and, then or. So if your expression is not completely parenthesized, or has the lower precedence than and. And you can look at various examples. And by the way, at any point anybody has a question, please feel free to interrupt me. We don't have to wait until the end of the talk. So generally, a modeling process begins with, okay, I have to understand the problem, of course, and we'll get to that. But once I understand the problem, I have to go basically through the following important stages. So the first stage is, let's identify the variables and the parameters. And let's take this out of the way. So this is interesting because I've talked with a lot of people who teach mathematical modeling, either in the continuous or in discrete case, it doesn't matter. The concept is difficult because we say, okay, the variables are those that change, and the parameters are kind of those things that also change. So what's because they do change, right? So that's a difficult concept for modeling students to understand. And we will come back to that, but it takes a while. So please, be aware of that if you haven't taught a mathematical modeling course. This is a place where a lot of time is spent. That if we talk about population growth, I mean, the most common example, that the population growth of the United States is not the same as the population growth per capita for Sweden, and so on and so forth. But those things change, but they kind of change in a different way, and we need to be able to explain this to our students. So after we've identified the variables and the parameters, we want to determine the dependencies, and that's what we refer to as the network of interaction. And then we have to determine the rules, what impacts what and in what way. And then specify the interaction dynamics. And for us, for the purposes of this tutorial, time is always discrete. All right, so identifying the model variables, we need to introduce some notation here. So for us, the variables are going to be denoted x1, x2, xn. Or when we get to gene regulation, we will use capital letters, A, B, C, M, L, just to be suggestive for what that variable stands for. And then the dependency diagram usually looks like this. So if you have, this is absolutely the same with differential equations, doesn't matter. You have to have that kind of network. And by the way, at this level, biologists are perfectly fine because they're so used to working with cartoons and links going here. No problem whatsoever at this level. So the rules of interaction, we have to say something like this. Those variables here will tell you how x1 will change. And those variables here are going to tell you how xn will change. And in general, if you have a fully connected, connections between any of the two nodes, you will definitely, you could have all of those variables impacting each one of the nodes. And the functions f tell you exactly what the nature of this interaction is. The important thing here is that in the wiring diagram, which I will call this from now on, you have a edge between xi and xj if the rule for the jth variable depends on xi. So this actually means, oh, in the rule for x2, I will have x3 present in the function f. And that's the intuitive way anyway, so that shouldn't be a problem. And then we want to incorporate the time. And the time is usually in those models done as, in an iterative fashion. So you have the state of the system at time t, and then you evaluate those functions, and you get the new states at time t plus 1. And various schemes for doing these updates are out there. So you could do this in various ways. For our purposes, for this morning, we're talking about deterministic synchronous update. So you take the old state for all variables, and then you use those old states for the whole round of updates. And you do not, in the process, you may have updated a variable that's used in the update of the next one. You don't use the new value. You use the old values until the full round of updates is performed. Now, often, now that we've introduced time, we want to talk about trajectories. We want to talk about the dynamics of the system. And we want to start with some examples. So if we have a system that has a wiring diagram like this, and the rules of interaction are determined this way, let's find the trajectory of this initial condition. And notice again here we have and we have or, and has higher precedence. So how do we do this? We really start with the initial conditions and we iterate. So that's really how it's done. And in this particular case, this is what things are going to look like. So you start with 0, 0, 0. x2 is 0, so x1 is going to take value 0. For x2, we have x1 or x3. Again, we come back to the initial condition. 0 or 1 is 1, so that gives you x2. And here we do x1 and x2, so 0 and 0 is 0, and then or 1, that gives you 1. And then you keep going. And of course, once you have hit this twice, you know that you have no point going anymore. So this is a fixed point. This is a steady state. And the nice thing about this is that we are space discrete, so we can actually look at all initial conditions, do this for every single one, and then put them all together. And so if you do it for this one, for instance, you're going to get a repeat kind of cycling between those two states of the system. So you have a limit cycle of length 2, and so on and so forth. And when you do this for the whole system, here we have just 3 variables. This is what gives you the space-state transition graph. And from the graph, you can look at disconnected components. You can find the steady states. You can look at the cycles and so on. All right, so just to recap, wiring diagram, update rules, state, space transition graph. Now, modeling using Boolean arithmetic to model biological systems is very old. So it was proposed by Kaufman in '69, and at that time, it wasn't really very popular as an idea. So people said, oh, come on, biology is so complex. You can't possibly do it with zeros and ones. Just forget about it. So it took some time, but now we seem to have a pretty good understanding that molecular biology is very close to digital. And genes are either expressed or not, proteins are either being made or not, signals are either sent or not, or transmitted or not. So this really seems to be the appropriate tool, and because it is so simple to get students to this level, I mean, notice that I haven't really used anything you cannot teach to a, I don't know, 9th grader, smart 5th grader probably. I mean, seriously. Things get more complicated when we get to the biology, because this is the biggest problem. And this is where mathematicians kind of hit a wall so many times, that you have to know the biology. I mean, I'm not saying you have to know all of it. You have to know enough to understand what your biology colleague is telling you to be able to translate into those rules, and then go back to her and say, does it make any sense? And she'll say no. You say, okay, let's do it again. But so that's one thing. Another thing, I was showing you this toy example for 3-node network. Real networks are much bigger. And so here you have a network that is of some importance to biology. I'm going to talk more about it on Friday if we want to talk about those types of things. But those networks, so as much as biologists are able to give us infinite level of detail, most of those networks are still kind of incomplete, because you will have the proteins, you will have pieces that are kind of well understood, and then you will have what people call concept nodes. That you go down this road and something happens, right? Proliferation, apoptosis. Yeah. And so we're not really going there. We're saying, okay, we're going to collapse all of this into a node, and that's what we are going to deal with for now. Okay. So those networks are huge, and the question of looking at the whole state-space transition graph is completely out of the question here, because the size of the graph grows exponentially. So other things need to happen. So we need to learn how to look at the rules, the update rules, the model, right? And figure out what the behavior of this model is going to be without simulating all trajectories. And this is not new, of course, because differential equations people deal with this problem all the time. You cannot solve it exactly. You have to figure out from the equations what the behavior will be. So nothing new here, just different type of approach. So our running example, because you have to start with an example, and ideally, of course, you'd want to look at many examples. Introducing an example, though, that is meaningful comes at a price, because you need to understand the biology. So given the time limitations here, our kind of basic example that we will use throughout for most of the discussion here is going to be the example, the simplest example of gene regulation that's out there, and this is the lactose opera. And I want you to keep in mind, while we're talking about this, that those two gentlemen figured it out without any of the tools that we have today. So they were introducing targeted mutations, and they were trying to see how that will impact the mechanism of the genes, and the whole mechanism of the lactose operon. And very deservedly, in my view, although nobody's asking, they got a Nobel Prize for this, because it really is, a remarkable work. But you will see that even in this context, even in this simplest example of gene regulation, things get to be pretty complex for a mathematician. This being said, they get pretty complex for systems that have thousands of genes, right, thousands of genes, hundreds of genes, probably more like it. And this is where modeling could help, because there is absolutely no possibility, I think, that for a more complex mechanism, the methods that they used to kind of understand the lactose operon would ever have a chance of working. So Matt later today is going to talk about reverse engineering, which basically is the question of, okay, if I observe this system on the macro level and I get measurements, how can I get the network? How can I get the interactions from the global behavior and then be able to study? Because even building the model may be quite challenging. Okay. So Robin,
righty. Well, I have to confess, when Raina first presented the Boolean model that we'll be developing shortly, I thought, oh no, that can't possibly work. I teach genetics. I teach the lac operon every fall. I've done it for the last 25 years. And when Raina sent me this model, I said, oh no, that can't work. And so I sat down and I worked my way through the zeros and the ones and the ands and the ors, and yes, it does work. It works beautifully. Yes, there are things to learn.
One converting power.
Yeah, absolutely. Yep, absolutely. Okay, so what I'd like to do is just provide a little context. And the organism, Escherichia coli, is the darling of molecular biologists. We've learned almost everything we know about the way cells function, at least initially from studying this organism. But I want to give you a little bit of background about how E. coli behaves and why this lac operon is interesting. So E. coli lives in our intestinal tracts. We're carrying around just an unimaginable number of these organisms. And what they eat depends on what we eat. So like all organisms, though, if they had a choice, right, if they could choose, they would choose glucose. And they do. If they have a mixed diet, if you will, they'll choose glucose. But they're only going to get glucose if we're eating something that contains glucose. And if we're not, then they need to be flexible. They need to be able to use whatever we're presenting them with. So if there is no glucose present, they will use other foods. But if a mixture of glucose and other sugars are present, they'll use the glucose first, and then they'll use the other materials. So let me go back to this slide. What Jacob and Monod did was to work out how the expression of the proteins necessary for using lactose was regulated. So there are two proteins here that are important, and we'll talk about why they're important in a second. Beta-galactosidase is an enzyme, and it breaks lactose down into glucose and galactose. So that's important. It also takes lactose and makes a slight rearrangement of the bonds into something called allolactose, which we'll see in a minute, and that'll be important. And then there's also the problem of how does the bacterium get the lactose inside itself to begin with? And that's what the second protein does. This is a transport protein called lactose permease. And if the bacteria has only lactose for food, it will make these two proteins, and then it can take up the lactose and then use it. But if there's no lactose present, it won't make those proteins. And if there's a mixture of glucose and lactose present, it'll use the glucose first and then turn on those proteins, make those proteins, and use the lactose. And this is a really intriguing thing. How can a minuscule, very simplest kind of life on the planet, how can a bacterium make such a sophisticated decision, right? It's got to see that the glucose is not present, its preferred food isn't there, and then it has to turn on the expression of these two important proteins and only do that under the appropriate conditions. It could turn them on all the time, right? But that would be really wasteful, right? If you make all of your proteins all of the time, you're wasting energy. And we know if you waste energy, you are not going to survive for long. And that is as true of bacteria as it is of us. Okay, so Jacob and Monod were interested in this property, the fact that they could turn on the expression of these proteins and use this lactose if it and only it was present, that they wouldn't turn on these proteins and waste energy in the presence of glucose. And so Jacob and Monod first worked out how these things were turned on simultaneously. It turns out there's another one turned on as well. That one doesn't have anything to do with our story, so we're going to ignore it. But by very careful mutation and then observation, they were able to figure out the order of these two genes and that last one there that we're ignoring. Okay? And also how that regulation occurred. So let's look. Oh, yes. This just tells us how permease works. It's in the plasma membrane of the E. coli. So if there's lactose out here and permease is present, the lactose will be imported into the cell. Okay, good. Once the lactose is inside the cell, beta-galactosidase, the enzyme, can do one of two things. It can break the lactose down into galactose and glucose. There's that wonderful energy source, that wonderful food. This one can be modified and turned into that one. A few more steps, right? So there's the food purpose of beta-galactosidase. And then the other thing that beta-galactosidase can do is just take this, rearrange a bond, and turn it into something very similar called allolactose. And that turns out to be important, as we'll see here. So if there's no lactose present, these proteins are not expressed, okay? Because the E. coli doesn't want to be wasteful, right? If we can talk about E. coli wanting anything. And the way that this is done is that before the first protein gene, there's an area called an operator. It's just a little stretch of DNA. And that little stretch of DNA is bound by a protein called the lac repressor. And if there's no lactose present, that lac repressor protein is made, it is active, and it binds to the DNA. And what that means is that RNA polymerase, the protein whose job it is to make a copy of that gene so that those proteins can be made, it can't do its job because there's a roadblock in the way. This massive protein is sitting there and keeping RNA polymerase from reading through and making a messenger RNA copy that can be then translated into protein. So if there's no lactose, then this thing is going to bind there, and we won't get beta-galactosidase or permease or that other one, transacetylase. Okay? All right. Sounds good. Straightforward. If lactose is present, how is the organism going to handle that? Well, if lactose is present, there's a small amount of permease in the plasma membrane all the time. There's a small amount of beta-galactosidase inside the cell all the time. So if lactose is present, some of it will get in. It will meet up with beta-galactosidase and get converted into allolactose. Just that slight rearrangement of the bonds. Allolactose, though, binds to the lac repressor. And when it does that, a conformational change occurs, and the shape of that protein is distorted, and it can't bind the DNA anymore. All right? So if it can't bind the DNA, then RNA polymerase is free to read through, make a message, and then this message will be translated into these proteins. Super. All right. So if lactose is present, we can make beta-galactosidase, permease, and transacetylase. But what if there's a mixture? What if we have both glucose and lactose present? Well, we, E. coli, does not want to use the lactose, because that takes up energy, if there's glucose present. So how does it handle that? That's a pretty complex question. Well, I only told you part of the story. In addition to the negative control, the repressor binding to the DNA and saying, you know, you shall not pass to the polymerase, there is a positive control mechanism as well. And this one is called the catabolite repression. All that means is that it is based upon the breakdown of glucose, glucose catabolism, glucose breakdown. Causes, through a variety of mechanisms which we can ignore here, causes the level of an important signaling molecule called cyclic AMP to be very low. And as long as glucose is being utilized by the cell, cyclic AMP levels stay very low. Okay? When all the glucose is gone, the cyclic AMP levels begin to rise. And when they have risen, what they do is bind to another protein called CAP, or catabolite activator protein, and that complex now binds to our lac operon DNA. And what it does is just bends the DNA a little bit and facilitates the binding of the polymerase. So now polymerase can bind, and if there's no lac repressor present, which there won't be because there's lactose, then the polymerase can read through and make our message, and then the cell can translate it and get these important proteins. Okay? So we have to worry about whether glucose is present or not. We have to worry about whether lactose is present or not. And we have to take into account both the negative repression, just go back, right? So this is a negative control mechanism, and then we also have to keep track of whether or not this positive control mechanism is working. Okay.
All right, so
you didn't know bacteria were so complicated.
Can I ask you a quick question?
Yes.
So in the case where neither glucose nor lactose is present, what's going on then?
They'll use other food sources.
What's going on with the machinery that you presented, though?
Oh, okay. So if no lactose is present, the lac repressor will be bound. Okay. So even if cyclic AMP levels are high, if that repressor is bound, nothing's going to get through.
Right.
Yeah.
And
So it'll be off. Okay.
So what will they be producing at that time?
Pardon me?
What will they be producing at that time?
Well, nothing from here. Okay. So they would be using other transport proteins, like say maybe maltose is present, so another sugar. They would mount a transport protein to pull that one in, and then they would metabolize that. Okay. Yeah. So this is just a limited picture of one particular set of genes that is well understood in one particular simple, well understood organism.
yeah,
Just
maybe,
a quick comment. If they run out of everything, they might also go into a dormant state and not do anything. So
Yes.
don't worry about the bacteria; they will be okay.
yeah. So this is the mathematician's understanding of this is that this is a lactose metabolism mechanism. So if lactose is there and glucose is not, the mechanism is on. And if we have neither lactose or glucose, it's off. If we have lactose and glucose, it's still off. So it only turns it on when the preferred food source is not there, but lactose is. So we have to go now to the modeling. So assuming we've understood the biology well, right, we have to go to the modeling stage. And typically, the phase of determining the variables is one where we want to make as much simplifications as possible that still preserve the biology behavior, right? We don't want to oversimplify, but we don't want to go with the most complex model that we can think of right away. So it's an iterative process the computer science people, and we have some here who are very well educated in this field, say computer programs are never finished, just released. And this is the same with mathematical models. So you will never make the perfect model, but if it's good enough, in the first stage, then you'll modify it, make it better, make it better, make it better. So our job here will be, okay, how do we select the variables from everything that that Robin told us? And here we have the lac y and the lac z. So this is, Robin, I always forget which one. This is the permease. Yeah, it's on the. This is on the membrane. So this is the transporter. And this is the beta-galactosidase. And so here they appear in more than one place because that kind of cartoon isn't meant to be a wiring diagram. It's meant to kind of show much more clearly the mechanism. So we'll say, okay, well, those guys here, it just, so this is a negative control, I'm sorry, here with the repressor. And this is the positive control here with the CAMP and CAP complex. And we will say, okay, well, first of all, let's start thinking about how to simplify. First of all, let's get rid of this amplification mechanism because, it seemed to come at the end, so we can probably ignore it for the moment. It's just amplifying. It kind of makes the whole process more efficient, but it doesn't seem to affect any of the regulators. And then we can say, all right, this is galactose, so we don't care about it because it's not part of the process. Then what else can we throw out? Okay, well, here I have that our lactose represses the repressor protein, which then cannot bind to the promoter. So this is a double negative. I can actually eliminate it and make it a positive link. So we'll get rid of that. Let's see. That leaves us with external glucose and external lactose, which we'll talk about. They seem to be fixed in time, and this is the thing where parameters versus variables kind of becomes important. It bothered me a lot. Why would I consider the external lactose and external glucose fixed for the purposes of the model? Until Robin told me that the lifespan of an E. coli is about 20 minutes. So what they do is they turn their operand mechanism on or off, but they don't have time to really go back and forth. And so we are not going to attempt to do that. So that leaves us with the other players. This is the messenger RNA for the lac gene, intracellular lactose that has gotten into the in the inside, transported by permease, beta-galactosidase, which on this slide is lac Z, our lactose, and permease. And so our first model will attempt to use those as parameters and those as variables. And so we say, okay, let's focus on this. And again, those will change their values in the course of time that we investigate within the lifespan. And within the lifespan of E. coli, it's very unlikely that the environment will change so much that we will go from different levels of glucose and lactose.
And we can just assume there's going to be so much glucose or so much lactose when it's present.
So the wiring diagram, now based on this understanding of the biology, looks like this. And I want to point out that we, as with every model, have to talk about assumptions. And now that we have eliminated a bunch of elements that we don't think are important for the first model that we want to create, let's make a few more. So let's actually assume that for those parameters, we have the values that are really of most interest to us. We do not have glucose and we have lactose, and see what will happen. And then we have to make some assumptions about the time step. And time is always a funny concept in finite dynamical systems, because in Boolean models in particular, because of course the time is discrete. So you have to decide whether your time step is a second or a minute or an hour. So here we assume that transcription translation require one step. So those processes will happen in one time step. And then degradation of the proteins and messenger RNA will also happen in one time step. This is not true for the lactose and all lactose. right? So those are different. So if I have lactose on this time step and I do not bring more from outside, lactose may still be available on the next time step. So it doesn't really go away at each time step. And everything that I'm going to say here refers to synchronous update. All right, so what are the update rules? And this is where it kind of gets really nice because you're doing modeling without really much mathematics. As long as your students have understood the biology, you can really restate the biology in terms of Boolean rules. So here's how it goes. You say, to make messenger RNA on the next time step, right? I want to have allolactose now, right? Because allolactose is the repressor, the inducer that will bind to the repressor, right? So I want to have allolactose now. And what does that say? I do not want beta-galactosidase. Why don't I want beta-galactosidase? Because that breaks allolactose into glucose and galactose, right? And so if beta-galactosidase is there, that breaking down will happen, okay? And I'm not in need of making messenger RNA, right? So you're saying, I need this, and I don't want that. And that's your first update rule. So this is, and of course, there are, I'm not saying this is the model. We will look at a series of models. But this is one possible way of looking at things.
I have a question.
Yeah.
Maybe a stupid question
Oh,
because
no.
my ODE background, but the update rules don't go with the diagram. Like,
Yeah, it's not one-to-one.
oh, okay.
Yeah, yeah. And we will get to that maybe Friday, because that's an interesting question. So if you look at that diagram, you can tell who's going to be here on the right-hand side for whom,
Okay.
but you cannot tell exactly how.
Okay.
Right.
Now,
And in some cases, and I should say here, you know, that's important, that you can incorporate some of those biology facts into the diagram. For instance, I've used red links, and I apologize, I should have mentioned this. I've used red edges here that have this kind of funny ending for negative impact. Right? So this is negative, so B impacts allolactose negatively because it breaks it down to glucose and galactose, right? And it also impacts lactose negatively because it cleaves it, parts of it, into allolactose. That's its role. Okay, but excellent question. Thank you for asking it. Because it's not one-to-one. We would love it to be one-to-one, and we will work to make it happen. Friday, if you want to talk about this, this will be a good topic to explore.
Ryan, why isn't there an arrow from D of M?
Not directly. Yeah.
Okay.
Yeah. So those are just the direct. And now, why do we have those links here? Because lactose can stay, right? If I have lactose on the next step, I could have lactose and so on. Yeah.
Sorry.
Oh.
Permease, this also take the lactose outside the colon?
No, it brings it in.
It's an egg.
Only brings it in.
Yeah, and we're imagining a situation where we have a huge amount of lactose on the outside and a small amount on the inside. So the net flow is going to be in.
Did you have a
I'm
question?
not sure on your rules why F of M is not just dependent upon A.
My first idea was, you know, let's make a bad model and see why it doesn't work, and then let's try to fix it, and so on and so forth. And it kind of worked because you didn't buy this model. Although I was trying to sell it as a, you know, that's a good model, so come on. So judging by the questions, you didn't buy it, which is good. And notice what happened here. So here I've been saying, you know, these are the assumptions, right? And so the assumptions are that those guys, the proteins and messenger RNA, they only last for one time step. So if I have it now and no new quantity is produced, it's going to be completely degraded down to base levels, right? I didn't say anything like this for the allolactose and for the lactose. So I didn't say, you know, beta-galactosidase will kind of break down the full amount in one time step. And so I was trying to do something with that, which which was saying, oh, if I have it, I don't want it to be broken down. But those are generally more stable. So some amount of this will be broken down, but some amount will remain so that it can bind to the repressor and messenger RNA can be made. So, by the way, if you run this thing, let me try to switch here. So this is a good kind of segue to the use of DVD. And DVD basically wants plain text. Here, the notation is consistent completely with what we've been using. The one thing you need to know about DVD is that negation is the twiddle, and that the expressions have to be fully parenthesized. So it doesn't really know precedence. It doesn't enforce it in any way. So if you don't have the expression fully parenthesized, it will stop you and it will let you do that. But this is the model that I had with the equation. So if I enter this into DVD and I make all the selections here, as we already talked about, then you can select whether you want the space space or a dependency graph, and then you click generate. And this is what you expect as an output here. And those links are going to take you to the respective graphs. So if we want to go to the state space graph, this is indeed a very bad model. Let's look at this and see why it's a bad model. The order of variables is, let me write it down, NIMBioS. L and P. So this state here says we have plenty of internal lactose and really nothing happens. Yeah.
Tell me again what you're calling this kind of representation. Did you say
State space transition, because the state space for the system is all the possible states, state as in this sequence of 5 zeros and ones that corresponds to the different combinations of zeros and ones that the 5 variables in this case could take. Right? And so here it tells me how the system transitions between this state and that state and any state. Okay. So this one says, gee, we have internal lactose, nothing's being made. Right? So this is not a good model biologically. Here we are saying the other component. We would have, so the first one sounds okay because, if we have beta-galactosidase and there is no permease, no lactose internally, that sounds okay. The second one is not good because we have lactose, but nothing's happening, right? So this is a very bad model, and I apologize for the way this happened, but obviously you did not really like that model because there were a lot of questions. Yeah, wait a minute, let's see what's going on here. So this is not what I intended, so I really apologize. So what I did intend to show you was, and let's go to laptop screen again, was to say, okay, well, let's fix that model, and I'm kind of halfway through fixing it here. So this was the original one. And we say, all right, so I want to say that if there is allolactose or lactose now, my time step is such that some amounts of this will still be available regardless of beta-galactosidase. So beta-galactosidase cannot break those down completely in one time step. Okay? And that's why here, that controversial point, which we had about having or not having links, I really do not want to have the link here. Come on. And I can say, all right, so if I have a lactose, that's good enough. I'll have messenger RNA made. If I change that, we can talk about this. We say, well, gee, do I want to have that clause here, or do I want to modify it? And again, this is good to have this discussion because this is part of the modeling process. I want to kind of briefly go over a much simpler model, which I haven't changed in the last 24 hours, and kind of talk about what we can do to now, if I want to incorporate all cases, as Matt was saying, we want to have zeros and ones, right, for all combinations for external lactose and external glucose. You have 2 parameters, and then you have 5 variables, so the size of the system becomes a little bit too big for demonstration. So you can actually reduce the number of variables, and here's how. You can look at what the permease and beta-galactosidase look like, and one is a tetramer, the other one is a monomer, and then you have the transcription rate the same because one messenger RNA codes for those 3 genes. Translation. There we go. And in that sense, you can say, well, gee, if I have one, I'll have the other because they are linked together. They're being made together. And also, because only a fraction of the lactose becomes allactose, I can actually collapse those 2 variables as well. And if I have lactose, I know that I'll have allactose. So we can make this reduction and say, let's collapse B and P into a single node E, and let's remove the variable A and assume that the variable A, and assume that L is the inducer. So I'll have to rephrase that as well. And so here we will have the reduced number of variables, 3 here, same parameters. And we can have a model that now incorporates the parameters. And so for this model, we're saying, okay, to have messenger RNA on the next step, I do not want external glucose. Right? I do not want it. And I want to have either internal or external lactose. And the internal lactose will do its thing right away. The external lactose, we always know that those small amounts of permease will bring enough lactose inside. So there is this implicit assumptions of trace amounts of the enzymes.
So just with that very first equation there, if you're starting with the assumption that external lactose is present, then the first thing just simply
Yes.
goes down.
Yeah. Yeah.
G.
So this is a good way to actually study, and again, I mean, this is a good way to analyze if you really are not just looking for fixed points, but to see what may happen in the system, to fix the nodes that you have some information about. So if I know there is no external glucose, I can fix that. This simplifies the whole thing, so I know this is 0, and I can only focus on the remaining sets of equations for the analysis. Right? So again, here for the lactose, I say I do not want to have this because internal lactose has no place to be in the cell if there is glucose. Right? And I need one of 2 things. Either I have external lactose, which this thing, which now stands for permease and beta-galactosidase altogether, this is going to bring this one in. Right? So if I have those together, that will make sure lactose gets into the cell. Or I want to have internal lactose, which is not broken down into glucose and galactose by the enzyme. And again, I mean, whether this will work or not, now I can guarantee that those are coded correctly. Because, again, this is when the operon is off. No enzymes, no messenger RNA, no internal lactose. And this is when the operon is going to be on. And here, because we have 4 different combinations for the parameters, we have to look at 4 different cases. And these are the cases. And we see that the operon really turns on only when it's supposed to be on, biologically. Right? It's off in all of the other cases. So no matter where you started, those rules kind of make sure that it either goes off under the appropriate biological conditions or it turns on. Okay, so this kind of passes the initial test as well. All right, so since the number of states grows exponentially, we want to know the fixed points, the special attractors. We want to cycles, all of this ahead of time. So how do we do that? We cannot do it by enumerating all the trajectories. So the fixed points are particularly nice because they don't depend on time. So we can really equate the values at at all times and just drop time from the picture and see what will happen there. So these are the questions that we want to look at. Can we determine the fixed points of a Boolean model from the equations? And the answer is yes. And we will show it in the next 5 minutes how. Can we determine the general properties of the Boolean system? And by general properties, I mean questions of this nature. And some of these questions are fairly complex. And David here is our resident expert. and he will, you know, if he may know the latest state of, you know, where those stand. Some of those questions are answered for special kinds of systems. So it's the same thing as we do in differential equations. And we say, okay, let's do it for this special class of functions. And another interesting question is, you know, can we use those Boolean models to exhibit bistability, which is a property in biology that is of significance. So let me briefly talk about how we can find fixed points. So here's a model that I'm not going to discuss, but a Boolean model of the lactose operand, much more complex. It has 9 variables. So we have 2 to the 9, that's 512, that gets kind of large. So how do we find the fixed points? Does it have fixed points? If it does, how do we find it? And the way to go about it is to translate the Boolean model into a polynomial model. And the transition here is really easy because the end translation into a polynomial is going to be this, so just the product, or would be this. And we can show that those are logically equivalent. So if you compare the truth tables, you can show that they're the same thing. And so finding the fixed points actually comes to solving a system of polynomial equations. And to do this, here is the translation. So for this model, I've used those rules. I've translated this model into a polynomial form. Right? So for each one of those operations, you apply those rules you've translated. And then to find the fixed points, you simply replace fm with m and so on. And so here is your system, and it's a system of polynomial equations. So now comes the point of asking, what does mathematics know about systems of polynomial equations? Can we solve systems of polynomial equations? We all know that we can solve linear systems of equations, right? Diagonalizing and so on and so forth. Well, it turns out there is a very nice thing that's called a Grosvenor basis. And for systems of this nature, you can use the Grosvenor basis approach to solve. And when you solve, you find 4 fixed points, and those 4 fixed points correspond exactly to the biology as we want the biology to be. But Grosvenor basis basically provide a way to translate a system of polynomial equations into a simpler form which correspond to the diagonalized version of a linear system of equations that we are so familiar with for Gaussian elimination, and then use a back substitution to produce all solutions. So this would be the Grosvenor basis is the analog of finding the Grosvenor basis and the appropriate diagonalized form would be the polynomial analog for doing Gaussian elimination on linear systems. And the reduced system is guaranteed to have a form that allows you to use this back substitution. And as I said, it can be used as a generalization of the Gaussian. Now, if you want to know more about why this works, because we are going to, in the afternoon, use SAGE for finding the Grosvenor basis. So there is a way to really use it as a black box approach and say, this is how you do it. If you know Gaussian elimination, you will be able to do that. If you want your students to open the black box and learn more about it, there are wonderful sources about it. Actually, it's one of the appendices that's included in your package for the afternoon. It's the appendix here. And if you have students who want to go into this black box, there's a lot of mathematics to be learned there. So you can talk about polynomial fields and ideals over polynomial fields, and Matt's going to talk about this. So this is a good project to do with research students. But if you just say, I need something to tell me how to reduce this system and guarantees that I'm not losing solutions, you can use it as a black box. And this is the SAGE interface that we will be using to find the Grosvenor basis. So if you have a SAGE account, you can simply log into it. If not, it takes 2 minutes to make a SAGE account. So what's left perhaps for next time? And it looks like, I really think that it will be my topic for the level 2 to talk about bistability, because Boolean networks, because we seem to have a lot of questions, and I'd like to continue with this and clean up a few things. So another question, so people say, okay, Boolean models are good, but they're so simple. Can they actually capture bistability? And what is bistability? Generally, bistability is trying to, looking at a system that has multiple steady states, but which steady state the system is going to go into depends on its history, basically. And so we are going to talk about the lac operon and its bistability property, and bistability in the continuous case looks like this. So we have those types of graphs for the bifurcation diagrams and things like that. But is it something that we can really capture using Boolean models? And the answer again is yes, we can. Can we model delays? So we talked about time being kind of funny. Okay, well, if I want to take a differential equation and then do a Boolean approximation of it, can I do that? And the answer is yes, we can do certain things there. I mean, I could show you just a kind of way in this direction. So how do we model intermediate concentrations? Because this is what bistability for the lactose operon actually is all about, that in the intermediate concentration you have bistability. Well, in the Boolean world, we're talking all or nothing. So how do we model intermediate concentrations? And I had included here some ideas of how this can be done. But I'll skip it. I'll talk about it next time. Here are the level 2 topic options for Friday. So at some point, after the hands-on activity this afternoon, we can begin to kind of narrow down the focus for what's going to happen with the Boolean network lecture on Friday. I definitely would like to touch upon bistability. This is going to be an interesting thing to do. How do we make the transition diagram and the rules one-to-one? Because now the rules determine the transition diagram, but not the other way.